coming. It's a pleasure to welcome you here. Today we have Professor Jesper Wengel, that is also a chair of the Danish Institute of Advanced Study. Jesper has a long career. He is a professor of bioorganic chemistry, director of the Nucleic Acid Center, and the Biomolecular Nanoscience Engineering Center. He's a professor here at the University of Denmark, but has been also privileged professor at the Copenhagen University, and associate professor before here. He has won a numerous number of awards. He has been director of the DG Center, Danish National Research Foundation for people that don't know Danish, and more recently also in ESC Advanced Grant. It's a pleasure to welcome Jesper, that is going to talk about plain chemistry on DNA and RNA.
Thank you very much, Francesco. Thank you very much for the invitation to come here today, and thank you very much for turning up in such large number. So I look forward to say some words about my background, a little bit history of my research, and then also on two ongoing research. I'll focus also on two ongoing research projects, at least some of the lines of it. So, as Francesco said, I'm now a chair in Danish IAS. I first heard about the idea of Danish IAS about 6-7 years ago. I think Francesco invited 3 persons from my institute to a dinner, and I have to admit I went away from that dinner thinking that this will never be realized. This is not going to happen. But I have to admit, if you take a little bit of Italian charm and a lot of energy, you need to be prepared what can happen. And if you on top of that add some Danish management style, Torbjørn, second director, I think we see that actually things can happen. And now it's going to be very exciting, I think, in the coming years to see how D-IAS hopefully will really bring advance to the whole University of Southern Denmark. This is at least my idea behind being part of it. Okay. So I'll tell you, if this works, I'll tell you about plain chemistry on DNA and RNA. So I'm a chemist, and I'm not primarily an observer of nature. I'm not primarily developing tools to look on nature and to try to answer some of the big questions in connection with nature. I early on decided I want to be a chemist because a chemist can be in the real concrete way creative, in that a chemist can generate new molecules. And this has always fascinated me. I'm not only studying what exists, but I'm actually also making some, I have the opportunity at least to generate something new. And I think this is a very fascinating aspect of chemistry. Then of course the question is what to generate. Maybe it's difficult for some of you to read here. It's written here, congratulations Jenkins, you've just synthesized artificial stupidity. And so of course you can generate many different things, many different new molecules. Some of them will have interesting properties and others will not. I early on decided I want to be a chemist, so in the lab, go to the lab and start creating new molecules. And for many, in particular those who are interested in organic chemistry, as I am, it is the aspect of possibly generating new medicines that is one attractive possibility. That was also really early on recognized in Odense, where I got my education. Here, this is Erik Bjerregaard Petersen, a few years younger than now, but still as active as that time. And he became my supervisor. He was working on development of new molecules that possibly could be used to treat HIV. This, in a way, was the start of what we have now, an education in pharmacy. I think this was one of the first real initiatives on medicinal chemistry, and Erik Bjerregaard has been a driver towards the development of the education in pharmacy that we have now. And also this, in a way, was the start for the Nucleic Acid Center, which was a center funded by the Danish National Research Foundation for 10 years, which focused on nucleic acids. And what we did at that time was at least the development towards working with nucleotides, so the building blocks of DNA and RNA. So this is what I will talk about, the building blocks of DNA and RNA. These are the molecules of life. Our genetic information is stored in DNA. It's like 4 Lego bricks. We have different ones. We have an A, a G, T, and a C Lego brick that can be put together in a strand. It's an oligonucleotide strand or a nucleic acid strand. And then a double helix can be formed in that the A Lego brick pairs with the T and the G pairs with the C. So a double helix can be formed, as you all know. And this double helix, which is where our genetic information is stored, that's DNA, where we have a hydrogen atom here in the sugar parts of the molecules. And RNA is the molecule where this, instead of hydrogen, is a hydroxy group, an OH group. So it's actually chemically, there's not much difference between DNA and RNA. So we have a sugar, we have a phosphate backbone, and we have a basis in DNA. And that forms the nice molecule of double-stranded DNA. I don't think it works. No, it doesn't. So, as I mentioned, medicinal chemistry is an important aspect for many, including for me. And this is the state of the art, also today, I think, of many medicinal chemistry in big pharma. It's not easy to find new drugs. It hasn't been for the last 20 years, not easy to find small molecule drugs. Now maybe you have heard that antibodies are coming up as new therapeutics, in particular against cancer. They have shown really interesting results. But the nice small molecules, easy to make, easy to use as a pill, it's very difficult. And this medicinal chemist, he has worked for 12 years. He has synthesized 10,000 compounds, and they are all inactive. No new drug has come out of that. It's a challenge. That's why Novo and in particular Lundbeck, they try to mingle around with their known molecules, do a little bit here and there, and hope to expand their billion-dollar sales for 2, 4, or 5 years more. It's simply difficult to get better drugs, in particular better drugs, than what we have today. And maybe these such DNA strands can be used as drugs. This idea has been around for more than 25 years. I've showed you the structure of a double-stranded DNA. When this information that is encoded in DNA is to be expressed, it goes via RNA into proteins. Proteins are the machines that do the functions that are encoded in the DNA. And this goes via RNA. And RNA, there are many different forms of RNA. There's a form, the messenger RNA, that directly codes for the proteins, and then there are many RNA forms that control the process of gene expression. And normally drugs, for example, small molecule drugs, also antibodies, they interact with proteins. What about developing drugs that look like DNA or RNA? Could that be possible? So here we have RNA. As I said, this encodes for a protein. If that protein is involved in a disease, for example, is overexpressed in cancer, maybe shutting down the biosynthesis of that protein could be one way to treat that cancer disease. And that possibly could be done by simply blocking the RNA. As I said, the DNA is 4 different bricks. That's the same with RNA. So, and we know that if we have a T here, we should have an A here. If we have a G here, we should have a C here. So it should be possible to design a complementary therapeutic oligonucleotide, a short DNA strand, 10 to 20 bases, no more. And in that way, shut down the expression of a given gene. That could be done with single-stranded antisense, it's called. It could be done with such a construct called a DNA sign. Or more recently, the last 10 years, short interfering RNAs have been developed. All of them have in common that one molecule, one strand here, one molecule here, one molecule here, binds to RNA using these Lego brick system, Watson-Crick base pairing, attacks the RNA, or either degrades the RNA, or simply sterically blocks translation from happening. Okay, simple principle, not quite so easy to do in practice. And you need chemists here. You need chemists to modify the structure. If I eat a piece of artificial DNA, it will be degraded within minutes and excreted via my kidneys, what remains, after a few minutes as well. It's impossible to use unmodified DNA. You need to chemically modify such that it becomes stable. There are enzymes that cleave nucleases. And you need to modify in a way such that the Lego brick rules are fulfilled, that they are able to target RNA very strongly. And also you need actually to be able to prepare the molecules. You need to have them in hand. We want to make drugs as material we can have in our hand. And I've been involved in that since I became assistant professor here, and also as I, in the late 90s, became full professor and stayed 4 years at University of Copenhagen, and also when I returned back actually as a professor here at SDU. A chemist can make different strange looking molecules. You can see in some we have made acyclic variants, we made some branched variants for various purposes, looking what will the properties be, trying to predict the properties, and unfortunately in most cases being disappointed. Oh, that didn't work quite as we expected. Let's try something else, or let's try to further modify. It's clearly an iterative process. But when we came to molecules that were bicyclic, like this one which we call LNA, we got some interesting results. And what is the reason for the interesting properties of LNA? Well, if you have, this is 2 different interpretations or structures of nucleic acid duplexes. This is a DNA-DNA, this is an RNA-RNA duplex. The sugars, that's the red part here. The sugar ring, they can flip. And they flip typically between 2 different forms, an RNA form and a DNA form. What if we do chemistry, introduces this bridge? We can make modeling showing that this efficiently will freeze or conformationally lock this ring into the RNA form. So we preform it as RNA. We were aiming for binding to RNA. So if a preformed strand is binding to RNA, there should be an entropic advantage in binding. It won't lose any degrees of freedom. It's not like a flexible molecule. No, it's a molecule I'm designed to bind to RNA. I'll do that. And so it did, and so it does. Without going into details here, we have very strong affinity to both DNA and RNA. If we take a DNA strand white, change it into LNA, or if we mix DNA and LNA or RNA and LNA. Bottom line here is just we have unprecedented binding affinity. Thousands of modifications have been made, and LNA was, when it was invented, about 20 years ago now, soon, it was the highest affinity binder, and it basically still is. There are now many analogues, but I don't think that any real advantages compared to LNA has been since identified when it comes to binding, at least. And it was, by the way, Paul Nielsen, who is sitting down here at the bottom, I see, who is now a professor also at FKF, who as PhD student in my group did the actual synthesis of the molecule. Okay, so we have looked into what happens with LNA, and again, I won't go into many details, but an interesting effect here. This is a DNA strand. Looking at the structure using Nmr Spectroscopy, we can look at the sugars. They are all DNA type. If the yellow ones here are DNA. If we change the yellow ones to LNA, okay, then you can see this one goes from 77 to 10. It goes from DNA to all RNA. So we have DNA nucleotides. They are swapped. Oh, suddenly, even though they don't know, they are not DNA anymore, they are RNA. So there is a conformational tuning of the DNA nucleotides. So there are some effects going on, and we have done a lot of work with collaborators internally here and externally on studying structurally what is happening with these molecules. How can we explain what's happening? What is happening with water around the DNA? Water is the most important molecule we have, and something happens with water around these duplexes. That's very important. So such studies we have been doing. And also, together with Birte Vester and Steve Douthwaite, who are sitting here as well, we early on did an experiment that I think was very convincing. And that was to take a DNA sign. This is this molecule here. It has been evolved to cleave RNA. What happens if we just take 2 of the nucleotides in this binding arm and 2 here and exchange from DNA black to LNA red? What happens? And we were targeting structured RNA, ribosomal RNA. Not easy. Double-stranded RNA is quite stable. You have to go in and open up the structure. Can we do that? We cannot do it, at least very inefficiently, with all DNA. You can see the green ones here. But with LNA, it's much more efficient. So suddenly we can target structured RNA. And as a chemist, I will, and I have for many years been believing that this tool is fantastic. Use it out there in the biology labs. And some have done that, but not as many as I had hoped for, because I think it's a very simple actually and quite enabling technology when it comes to targeting RNA. Very simple chemical modification has a strong effect on the performance of these artificial molecules. As I said, Paul developed the synthesis. I showed this. I decided to put this up just to show you that the chemists need to work in the lab. This is an optimized synthesis, basically the same steps as Paul developed. It's a quite efficient synthesis. It's a 12-step, a high overall yield, and that has been optimized in a Danish company called Exicon at that time. So we need to be able to synthesize the molecules. Very important. Okay, this is supposed to be a very confusing slide. So that was a little bit about the background. So we were able to set up the Nucleic Acid Center here in Odense. BMB and FKF were involved. We were 5 research groups. And the work that was done early there and a little bit before also led to, with LNA, to Santaris Pharma and Exicon. Both companies have now been sold to foreign companies. Still about 200 employees in Denmark and a number abroad in different companies. Drugs are under development with LNA, based on LNA. Then we went on to actually take part in EU project and also in Højteknologifonds, Innovation Foundation type projects. And also a small company that I co-founded together with my research group from NAC was part of that. So this was, you can say, innovation focused. In that project, we started some work on what is called aptamers. It's something about molecular evolution that actually led directly from Højteknologifonden back, one can say, to an ERC advanced grant. And also other companies came out of it, and now we have what I will return to, Danish Center, a center funded by the Willem Foundation for basic research. So within this field, it doesn't really give much sense to speak about basic research, applied, strategic, innovation. It all is very closely intermingled at least. And you can actually go from innovation, product development, back to basic research. There are researchers today that use LNA to answer very basic questions on RNA. So things go back and forth here in this biotech chemical biology field, as in many other fields, but certainly not all fields. Okay. As I mentioned that LNA is involved in this type here and is in clinical trials. Hopefully drugs will approve, but of course this is a question mark yet. There was another strategy, small interfering RNA, and just to let you know, we also synthesized this molecule. This is not locked nucleic acid, this is unlocked nucleic acid. And I won't go into details, but both of them can be considered RNA mimics. They are both actually quite unique in the field in that they can be mixed with DNA and RNA. So researchers can pick, I want to have a little bit stronger affinity to RNA, I put in one LNA, a little bit more, two, a little bit less, I put in one UNA, a little bit less, two UNAs, etc. And both compounds are commercially available. This is also very important because otherwise nobody can use it. So I enjoy reading when researchers use these tools to get answers to their basic questions. That's only possible because it's commercially available. Okay. So just 3 slides, I think, about siRNA. So that's this part here. Sorry, this part here. This is also a very active field, used to answer many basic questions, and also used for drug development. And UNA is useful there for a specific reason. You can put it in this position of one of the 2 RNA strands. These are 21 strands. If you put a UNA here, you get high potency, but also reduced off-target effects. Maybe, at least some of you have heard about siRNA to down-regulate the expression of a gene. Yes, but besides that, maybe 150 other genes are also mostly down-regulated. It's not very selective. If you put in a UNA, this number is not 150, it's maybe 15. That is a significant improvement. And the reason is that there are 2 strands. Now we take only the blue strand. This can work as an siRNA, it can work as a microRNA, or it can work as a seed microRNA and as a full microRNA. It's a bit technical, but for gene silencing we only want this. And by putting UNA here, we can actually achieve that. And that can be seen, let's go directly to this one here. You can see here, no effect as microRNA seed, only effect as gene silencing. Without going into details, just showing you that this is how chemists work. They try to change the molecules such that hopefully the properties will change. And here we see a much more selective gene silencing than without this little change. It's just, in this big molecule, it's just one bond here that has been cleaved. Instead of having the cyclic ribose, it's now an acyclic ribose. UNA is also in clinical trials. There are two American companies, Arrowhead and Arbutus. They have UNA-modified siRNAs in clinical trials, phase 1 and phase 2. Okay, so now I'll move on, again without too many details, to describe two ongoing projects in my group. One is also on medicinal chemistry of oligonucleotides, and the other one is a BioNEC, Center for Nanoscale Biomolecular Engineering project on really to see, can we use oligonucleotides to scaffold peptide strands or to scaffold protein mimics in a very basic sense. So first, a little bit about can we do some medicinal chemistry on oligonucleotides. And there are 4 drugs approved so far in this class of compounds. So short DNA type strands. This one is not sold anymore. This one neither. This one is sold to reduce cholesterol in a very narrow patient group. It inhibits the gene that produces cholesterol. It's sold a little bit. This one is a chemically quite radical molecule called a morpholino oligonucleotide. It has just been approved. It's going to be sold soon, and the problem is it doesn't really work. So the success has been limited. There are obvious problems in this field when it comes to having such big molecules go into cells. That's a major problem. There are different chemistries that are used. You can see the leading US company is using this molecule here. It's the same as LNA, but has a methyl group here. And that's for license reasons. They couldn't get along with the Danish company, and therefore they had to, in one way or the other, get their freedom to operate. But they are advancing this molecule very nicely. But when you think about thousands of modifications being made, it's quite a low number that has actually moved into clinical trials. And they have some specific challenges. I won't go into details, but instead of phosphorus, they have thiophosphorus in their linkages, and that gives toxicities. I just listed up here. I said excretion is extremely rapid. This is a major problem. And therefore, binding to blood, to various proteins in the blood is engineered, and that is typically done doing these thiophosphate linkages. And they generate diastereoisomeric mixtures, so instead of one 16-mer, you actually have more than 32,000 different 16-mers in the drugs that are being used. At phosphorus, different stereochemistry. And that gives, at least adds to the toxicity in various ways. So there is a desire to try to avoid these thiophosphate linkages. LNA is also difficult because normally here there is actually a sulfur. I think there's a lot to do with medicinal chemistry to improve the so-called pharmacokinetic and off-target effects. It's not just injecting a drug. It needs to be dissolved right. It needs to go into the right organ, to be taken up into the right cells, and if that's the action, and as it is for these type of molecules, and within the cells, they need to be able to do their action. So it's a long journey. It's not easy, and it might work the wrong places to give tox effects. So there's a lot that could be tried, and I think that this best could be done in biotech or therapeutic companies. but they just don't do it because they haven't got time for it. And recently I have realized that this is how it is. And I think there is so much more that could be done that I have decided to try to do at least a little bit of it. And it takes time. And you need to work with biological collaborators and even mice. And none of them are easy to cope with. Mice are now well known. So it just takes time, of course. One molecule that we worked with is lipophilic amino LNAs. You can see it looks like LNA, but it has a nitrogen here and then a fatty acid here. That still allows binding to RNA, and the question is, does it actually improve the properties of the molecule? Could it give better uptake than LNA? Better lifetime in blood? That would be good. We have looked at binding to albumin. Albumin is the dominating protein in blood. And for example, the new long-lasting insulin from Novo Nordisk has fatty acid attached in order to bind to albumin, to make it long-lasting. What happens if we do the same trick with oligonucleotides? Here is no binding to albumin. It's difficult for you to see. There is no palmitoyl, there is no fatty acid in the oligo. Neither PO nor PS. PO is the phosphate, PS is the thiophosphate. One doesn't help. No binding to albumin. This is albumin autofluorescent. With two of these palmitoyls, suddenly there is a very good binding to albumin. And the same is the case if the two palmitoyl fatty acid chains are positioned at the end of the molecules. So there is now significant binding to albumin. This could be good, because that could maybe give a longer half-life in blood. Do the molecules work? Yes, they do work. This is an example where we're looking at gene knockdown. So we want to be down here for various different constructs against a given target. These two signals here are for these palmitoyl fatty acid oligos. So yes, they do work in cell culture. So one takes cells and add the oligos and see if they work. That's good. Are they taken up even without transfection agents? Yes, they are. And again, I won't go into details, but some of the bars here are for the palmitoyls. They are taken up into the cells, just to show you how we have to work with biologists on this. Are they stable? Do they stay in blood? This is after injection into mice. Oops. And oh, I don't know why that doesn't come up. Then you have to believe me that this is the standard LNA PS thiophosphate oligo. It has a half time of about 20 minutes, less than 20 minutes in blood. It's here. Much more stable presence in blood for these dipalmitoylated oligonucleotides. So yes, we can increase plasma half-life significantly. Does that have an effect with the distribution into organs? And yes, it has. I don't know why it doesn't show up. Strange, but we have the kidney and liver distribution here for LNA gammas. Very strange. This one here is for the palmitoyl derivative, and you can see that it goes much less into kidney and much more into liver. It also goes more into spleen. So there is a change in biodistribution. We don't lose it via the kidney rapidly. So things like studies like that have to be performed. And we also have seen knockdown in the liver against a target. I can't show you that right now, but they do work. To what extent they are better than normal, state-of-the-art antisense oligonucleotides will have to be shown. but some of these properties that they work in cell, that they do show extended circulation time, and that even for phosphate, we can probably avoid the thiophosphate. That could be very important because toxicity really kills many drugs. That's promising. So we are continuing working on this with different partners to see what kind of properties we can engineer by making this or similar changes. We make a range of different changes. We also put in positive charges here in order, so to say, to make the molecule less negative. Because it's not easy for a fully charged molecule to go through a lipid membrane when it has to go into cells. Okay. That was a little bit how we work with medicinal chemistry of oligonucleotides. Then what about devising a new concept for artificial protein mimics? That will take maybe, what's the time now? Yeah, it will take 5 to 10 minutes, and then we will be finished here. So this is done within BioNEC. BioNEC is Biomolecular Nanoscale Engineering Center. It's funded by the Willem Foundation for 5 years. And we have 4 groups involved. One group that knows about nucleic acids. Knud Jensen in Copenhagen knows about peptide chemistry and carbohydrate chemistry. Stefan Vogel here at FKF knows about lipid chemistry, also about peptides and nucleic acids. And then Jørgen Kjems in Aarhus, who is he's a molecular biologist, a lot of nano capability and instrumentation and nanotechnology, biotechnology in general. So we tried to join forces with respect to knowledge on the major biomolecules of life, lipid, carbohydrates, proteins, and oligonucleotides. And then we wanted to study new hybrid materials, and at the end also our idea is to bring these nanoscale engineering activities via chemistry to life, to biology. Our first real full BioNEC project has been a project on assembly of artificial 3-helix alpha helical protein mimics. So now we don't work exclusively with nucleic acids, we also have peptides. And our idea is to use the oligonucleotides to scaffold the peptides. It's very easy, using the LIGO principles, to get the oligos to assemble in water, in salt, and then can we use that to enforce peptide units to come together, hopefully to make artificial proteins with interesting properties. Synthesis has been to be done, and Harvey, a very skilled postdoc in my lab, has devised some very efficient methods for synthesis. It's a fairly long peptide coupled with different oligonucleotides. The isolated yields are impressive. It's difficult to do these fusions with such molecules, and we now have a method where we can do that, where Harvey can do that in an efficient way. What happens with the molecules? Is there an effect of having this is a duplex. This is a duplex with 1 peptide, this is a duplex with 2 peptides. Do the peptides react on the presence of the covalent attachment of the oligonucleotides and the assembly process? Or is it the peptides that actually has some information to transfer to the oligos? Well, we do some melting studies here, and at least you can see the numbers are different. So if we have a peptide to each strand, the temperature is higher. So the presence of the 2 peptides, the interactions there, has an effect on the stability of the oligonucleotide. So there is an information transfer in that direction from the alpha helical peptide down to the oligo. this peptide sequence has a tendency to form a coiled structure. What about 3 strands? We can put on a 3rd strand on a duplex in a so-called triple helix. And if we go down here with all 3, we can see that we have a second transition here. This is a melting transition, a triplex transition. So you have to believe me when I say that yes, the 3 peptides, they have a stabilizing effect on the oligo segment. A little bit different what we expected. We expected more than that the oligo would exert an effect in this direction. And it does, but I'll come back to that. But the peptides also, by their tendency to self-assemble, has an effect, a stabilizing effect in that direction. We have looked at the alpha helicity of the peptide. So does it look like that, or is it more like that? And here we have this is a so-called CD spectrum. We have the 3 oligonucleotide, the 3 DNA strands here, has a signal here. The peptide alone has a signal here. If we mix them without chemical linkage, we have an additive effect, so the sum comes here. But when we look at the alpha helicity of the peptide for the conjugate, for this structure here, it's very pronounced. And as I understand from our Copenhagen group, it's unprecedented. So there is a transfer of helicity from the oligonucleotide part via this linker to the peptide part, I believe. So yes, there is a crosstalk in this direction, but also a crosstalk in this direction. And we have looked at the structure. At high concentration, we believe that we have a dimer of trimer. So whereas at the lower concentration, we have the expected monomer of trimer. These are 3 molecules that come together. Therefore, we call this a monomer of trimer. This is a dimer of trimer, where 2 of these go together. And also TEM shows the same, that at low concentration we have the monomer, whereas at higher concentration we have a trimer. So this project has been possible only because of the joint capabilities within BioNEC. And we are now working in different directions to continue our studies. This is an example where we have shortened the peptide. Again, Harvey developed some very efficient synthesis, high yields of the conjugates in a very pure form. This is how chemists also work. They isolate the compound and then they prove what it is and that it is actually pure. And again, we do see synergy, not so much when it comes to binding as it is shown here, where we look at just the duplex. But when it comes to the triplex, we again see a stabilizing effect of having the shorter peptides together onto the oligo part. This is just to remind you, I've skipped that one. So this is the CD for the shorter peptide. And the peptide in itself shows no tendency to form the coil, only in the presence of the oligos. When conjugated to the oligos, there's actually alpha helicity. So we enforce the peptide to form a structure, the 2 peptide strands to form this coil structure, which forms is a structural element of many proteins by this attachment and by this design and self-assembly. And we believe here that even at high concentration, we exclusively have the monomer present. So we are continuing that work in various directions. We work with insulin at present in various other constitutions. And we are planning to move in the so-called dynamic combinatorial direction, where we have encoded oligonucleotides that contain different peptides, and then see how they will assemble when we ask a different task to them, for example, binding to a given cell. So these are some of the directions that we are moving in. Okay, so with that I would like to start. And people have been involved with the medicinal chemistry aspects, and there are people associated with was and still is, I would say, the Nucleic Acid Center. You are still here because of the nuclease experiments, and Lykke as well. I can see it sitting over here. So thank you for collaboration on that. And also collaborators within BioNec, and also in fact for the therapeutic work from Aarhus and Copenhagen, which I have listed here. It's okay. So I hope you at least got an idea of the kind of work that I'm involved in. And once again, thank you very much for turning up here now. Thank you very much.
Thank you, Jesper, for a very nice lecture. Actually, I should say this is the first lecture I heard in 10 years I came here from you. And so in that case, I think that was very lucky for me. Questions?
It's a really interesting talk, thanks. And I was glad you got into the aspects of sometimes reduced specificity when you're doing the siRNA knockouts and microRNA knockouts. And that probably is even, lack of specificity is probably an even bigger problem with these CRISPR-Cas9 systems that are being used now to make knockouts. I read somewhere that they're knocking out all the retroviral sequences in the pig so they can use organs for human transplants. And they are probably not very precise, 100% precise. And I was just wondering if you've been looking at the CRISPR-Cas9 systems and getting involved in those in some way.
Well, we have been discussing, certainly, and Per Trolle has also said to me many times, you should do this. But I said, I'm sure that ISIS have already done it, and others have. There have been now publications on the use of LNA in these kind of systems, and they sometimes improve activity. I've suggested chemists that we should do this with you exactly to improve specificity of the system.
Yeah, because activity, you know, to get that to go up, especially with LNA, but it's specificity that's the main point.
Yeah, but we haven't started. I really think we should start that, and maybe the activation barrier for him is a little bit too high. So I'll be very interested in trying that. But exactly, it should be to tackle the selectivity.
Yeah, no, that'd be good to get your input on that.
Fully agree. It's a good point.
So we also have a question.
LNA is a real invention, as I see it. It's a new molecule with new possibilities, and I see it as a real invention. And it was done 20 years ago now, you said. And you also said that despite that, no real clinical testing materials based on LNA have been performed yet. Is that correct?
No. What I at least tried to say was that it is in several clinical trials. It has been in several clinical trials that have been closed. It also still is in itself, but also with the methyl group attached.
Yes.
It is in several tests, including I think 1 or 2 phase 3 trials. I'm very convinced that a drug will come out of with the methyl group attached in the not so far future. So it's the LNA but with a methyl group.
Yeah, I saw that, but I'm wondering about it. Why does it take so long? I mean, it's clear this is a new tool that can do things that no other molecules could do before that. And why does it take 20 years? You said 20 years, yes, it's clinical testing, but it's not really out there. You can buy it as a little jar or something like that. It's not out there yet. And I was just wondering why is that the case? Because is there something here about them invented at the wrong place at the wrong time, or by the wrong people, or what's happening here? Because in other circumstances, I would say a real invention like this had been out there for a long time. So is there something in the history you think this because I've been following it since it was invented? I've been wondering why we still waiting for this thing in the jar to come out.
It's a very interesting question. I've been asking that to myself many times also. You can buy it, but as a reagent, not as a drug. Paul and I discussed, of course, what should we do 20 years ago. And one option was to go to this company that now has the methyl. US company, I knew the people, go there and say, you can get it. Pay us a little bit. It was I thought it would be nice to first of all, I wanted to publish, I wanted to forward my academic career. That was my focus. But then it is so obviously interesting. And then we went to a small Danish company. I thought it would be nice to do it in Denmark. And we agreed that yes, jointly wrote a patent application. Paul and I got a little bit of money, not much I have to say, unfortunately. And and then they were to take it. I wanted to be in academics. And we agreed what we should do was to buy a big amount of LNA and buy a helicopter, fly around the world and give it to anybody who wanted to try it for free. What happened was the exact opposite, that a German company got in, they got the rights to produce it, and they wanted to earn money, of course. So already from that stage, the price went up. That was one. I don't know how to get around that, but that happened. Then Santaris was formed, a Danish drug development company, with people who, that was a daughter company of Exicon in the beginning, with people who were not knowing what drug development was all about. This is the second lesson, that you cannot develop these drugs, I think, in biotech companies. They don't know what to do with them. I'm fully convinced if Santaris would have been pragmatic with respect to a license to ISIS. ISIS, they work with all the big pharma companies. And they are 300 people. They have 30 compounds in clinical trials, 10 in phase 3. With 300 people. It's unseen. So if they had had the opportunity to work with LNA from the beginning, either if Paul and I had gone to them, or if Santaris had, then it would have been out there. That's my personal, maybe 2 or 3 years ago. It takes, of course, these 12 years or so to develop a drug. They have done so much chemistry to get around these patents because the stubborn people at Santaris wouldn't give them a license. That has happened, in my opinion. With UNA, I decided this is a molecule that doesn't at all has the prospects of LNA. I decided I don't want to interfere with those incompetent people anymore. I'll do it myself. Okay. So I made Ribotask and we made some licenses. Then, of course, I made other mistakes instead. But at least that molecule with much less prospects or possibilities is at present almost as far in the process as LNA. And this is in a way very sad that this is the case. So you're right.
So this is a question which is goes outside this.
No, actually it's sort of a follow-up question. First of all, great talk, but a couple of years ago there were a lot of pharmaceutical companies that were dropping their RNA-based therapeutics, Merck for example. And there was a lot of negativity in the field in general about RNA drugs. Do you feel it's bounced back, or what has happened since?
Yeah, I think the field has bounced back. And in particular, the single-stranded antisense has bounced back. Yep. I think for single-stranded antisense, it was a great luck that RNAi came up, because that again turned on the interest of the big pharma companies. And now they are more or less all of them changing from siRNA to single-stranded antisense, and in particular to these LNA or methyl LNA-based systems, because you can test without transfection agents.
Yep.
So there is a much better transferability from in vitro to in vivo. than if you use siRNA, where you have to mix in all these lipids, and that they don't like to do that. That's also why Roche has, I think, bought Centaurus. Patent-wise, there was not really a reason to do it, because the LNA patents expire now in about a year's time. But I think it was anonymous and things like that. But so I hope that now that will change also. And there are US companies that work with LNA based on licenses from Centaurus, older licenses, that are doing quite well. So I think the field has bounced back, but also I think the field needs a success. Those 4 drugs that either don't work or don't sell is not the best advertisement.
It's a great story. So I'm also coming from the biology side and have more questions about the specificity, because the other level of specificity, as you also find out, was hitting the right organs and hitting the right cells and right organs. Is it trivial? It seemed like quite easy to actually trace your LNA or modified LNAs to specific tissues. Is that doable in vivo, also with
fluorescent scanners, for example,
okay.
Or you look at knockdown in genes that are expressed everywhere.
Okay, and have you made any attempts to actually direct not only to the liver, but specific cells of the liver or other organs, tumor cells, for instance?
We have done something to target tumor cells, and we also have some I think interesting results that we try to push now with aptamers that are targeting tumors. But it's only about 1 year ago I decided I'll go back to this and try to see what I can do, because really the biotech and big pharma is too conservative here. And furthermore, we don't know all about that. We don't know all the results, in particular not the negative results. So I think we have to do it. So it is not trivial. It is difficult to target specific organs, but there's a lot of activities going on towards that at the moment.
Because I think even ISIS, or I think they're called now for good reasons,
Ionis, yes, that's true.
They have issues with blood clotting or platelets.
They have, They have,
So.
Yeah, they have. Tox issue is the second. Some of these tox issues are concerned with these thiophosphates, but others are not. So that's true. I think that's another major issue.
Further questions? Well, if not, let's thank Jesper.